Hello again, chapter 19, part 2. Okay, so overview of mitosis. I've said this a couple of times. You're probably bored of this. Here you have your unreplicated chromosome. It's a chromosome because it's condensed DNA. It has a centromere here. Then we are going to essentially copy that genetic material to have two copies. Again, attached by the centromere. And we call this the sister chromatid. Okay, so a little bit of an overview of what we're trying to do here in mitosis. So here's your original cell. What you should know here in the purple is our nucleus. Here's that nucleoid, kind of getting out of the way. And then you also see that we have some chromosomes here. Now, I'm just going to go ahead and assign them. The red ones are from Dad. The blue ones are from Mom. Now notice that one of each of these red and blue ones are longer and one's shorter. So I'm going to refer to these longer ones as homologous. Chromosome 1 from Mom, chromosome 1 from Dad. Maybe this one's chromosome 12 from Dad, chromosome 12 from Mom. We have two copies of chromosome 1 and 12, so we refer to that as being diploid, 2N. N is the number of different chromosomes. So we have a chromosome 1 and a 12, so 2. And we have one from Mom and Dad, times 2. So this means that the total chromosome number for this diploid individual is 4. Now, in humans, N is 23. We have 22 autosomes, and then we have sex chromosomes. So that's 23, and you have one of each of those from Mom and one of each of those from Dad. So 2 times 23 is 46. What we see in mitosis is that, remember, we are going to replicate those chromosomes. And now we're going to have sister chromatids. Okay? And there's our two chromosomes, chromosome 1 and chromosome 12, and the two copies. But now they're in the duplicated state. So those are sister chromatids. At the end of mitosis, we are going to have separated the two copies of the sister chromatid for each of these. Okay, so now you see in the cell, you have one copy of each of the chromosomes, just like the original cell. What's different here is that we actually have two cells. So the one cell made two cells, genetically identical. So let's talk about the phases of mitosis. Okay, they flow one into the other, to the other, to the other, and it's just constantly going on. As humans and curious people, we like to study these things and name it, but in life, it's just constantly going on. So let's take a look at these different, as they call them, stages of mitosis. Okay, so this should look somewhat familiar to you now. The nucleus. These are condensed chromosomes. They're not quite completely condensed yet, so we will call this early prophase. Also, in early prophase, we're going to see that the nuclear membrane is going to begin to break up. It's literally in the way of cell division, or what we call karyokinesis. And here, in this particular situation, we're referring to the division of the nucleus itself. So the nuclear membrane breaks up or fragments, and now it's just kind of around the cell. The other thing that you're going to notice is this thing is part of a microtubule organizing center. We can call it the spindle, but essentially what we have here is some centrioles and some microtubules that are going to play a role in pulling apart the copies. Okay, so prophase, now we see the nuclear membrane is fragmented, and we go ahead and see that, oh yeah, there are those sister chromatids. They are condensed and we can see them. And the spindle is starting to spread out. Prometaphase, which I don't really care that you know that, but pro means early metaphase. Essentially now we see that these things are kind of starting to move around. And what are they doing? They're actually going to line up at the center of the cell. Every sister chromatid is on its own. You notice that. X is down the center of the cell. The center of the cell is called the equator, and meta means middle. So one way to remember metaphase is that the chromosomes are in the middle of the cell. And this spindle is essentially now growing so that we have these microtubules connected to each sister chromatid. And those microtubules essentially are going to shorten and shorten and shorten and shorten, pulling on the centromere. So remember, the centromere was the piece of protein that kept the two sisters together. So by attaching microtubules from opposite directions and pulling them and pulling the centromere, then we're pulling the copies apart, which is brilliant, right? We had one cell with the DNA in the nucleus, and we fragmented the nucleus. We replicated and condensed the chromosomes, and then now we're forming the spindle and pulling the copies apart. So we see that here, and when we pull the copies apart, we call that anaphase. And it continues until essentially these chromosomes are separated from one another. The nuclear membrane is going to reform from those fragments that was hanging out in the cytoplasm. And then there's a pinching that's going to occur. So when we divide the nucleus, it's referred to as karyokinesis. When we divide the cytoplasm, that's called cytokinesis. So in animal cells, essentially what they're going to do is there's going to be a point of constriction and it's going to pinch apart. Now what we have are two individual cells. Brilliant. Okay, let's now talk about the other kind of cell division and contrast it as we go with mitosis. This is called meiosis. This is going to be the division of the cells that are necessary for sexual reproduction. So it's going to occur in what we call oogenesis, or the making of eggs, and spermatogenesis, which is the making of sperm. It's going to look different, but the principles are pretty much the same. So think about the principles that you remember from mitosis. Now, what we're going to do is we're going to take those cells that are diploid, right? You had the two copies, or the two sets of chromosomes, 1 through 23 from mom and dad. And now for you to make your own individual, unique sex cells, you need to go through a different kind of cell division, because you only can contribute just one of the sets of chromosomes to your offspring. So in meiosis, we go from diploid, or two sets of chromosomes, to haploid, which is just one set. In mitosis, we made two cells as a result. In meiosis, we're going to have actually meiosis 1 and a meiosis 2. So really a two-step division. And in the end, we make up to 4 types of haploid daughter cells, which in this case are either a sperm or an egg plus her polar bodies. Okay, I think we talked about homologous chromosomes, so pause the slide if you need to see it again. Okay, the one huge thing that happens on meiosis, so that your sex cells aren't exactly chromosomes that your parents gave you, you have to make your own kind of mixture of chromosomes or traits on the chromosome. Genes are the same, whatever that's coding for, but the variants, the alleles, are different. So in meiosis 1, there is a thing that's called crossing over. So essentially what we have, the homologous chromosomes, so one of these is from mom and one from dad. Before we said red was from dad, blue was from mom. And you're going to see they literally make connections, and pieces of the homologous chromosome from mom and or dad exchange with each other. So now we see that the resulting chromatids are not exactly what they were to begin with, which is awesome. So now we're going to increase genetic variability. So let's take a look at meiosis. I don't want you to know all the phases of mitosis. I want you to be able to contrast meiosis with mitosis in general. But what we have here is, again, we're going to replicate all the DNA just like you did with mitosis. Now what we're going to see is that the homologous chromosomes pair up, and they're going to do that thing crossing over. So the synapses, the connections, and the crossing over. Then we're going to go through the two-step division, so we get singles. Okay, these singles, or these haploid cells, are not going to be like the original cell. First off, they only have one copy of each of those chromosomes. Chromosome 1 and 12 we said before. So N equals 2. There's not two sets of each of these chromosomes. To slow down a bit and to show you more specifically, in meiosis, what's going to happen is we're going to have the crossing over here. Happens in prophase 1. Now take a look. Metaphase still kind of means the same thing. These chromosomes are really, they're called sister chromatids. And in meiosis, we call it homologous sister chromatids, are lined up at the equator. And we pull one of those X's, or the entire sister chromatid, in one direction, and the homolog goes in the opposite direction. But notice at the end, we still have two copies. So that's why meiosis 1 has to go through meiosis 2. So the meiosis 2 is going to look like mitosis. We take the sister chromatid, we put it on the equator, we shorten the microtubules, we split apart the sister chromatid, and now we have singles. Okay? And you see that there's up to 4 daughter cells. So what we see here is that those sex cells are going to be unique. Every single one is unique and different from every other one that you're going to make. And the reason why they're doing this is so that in the event that fertilization occurs and you have you're going to have a child, that we can restore that diploid nature. And now you've got different genetic characteristics that are unique to you. Okay, so in spermatogenesis, you see the end result is 4 haploid sperm. Count them, 1, 2, 3, 4. In oogenesis, so when we make eggs, weird little thing happens. First off, a female baby, all of her eggs are stuck in meiosis 1, right here. And if that egg is fertilized, of course, puberty or after, then this cell will finish meiosis 1 and it makes a little polar body. Okay, essentially it's a little tiny cell, nucleus, hardly any cytoplasm, and a plasma membrane. Okay, this polar body gave her extra cytoplasm to this cell that's destined to be the egg. After meiosis 2, we make another polar body. So essentially what you're going to see is you're going to have one gigantic egg, and in the end you have 3 polar bodies. Okay, genetic recombination is the significance of meiosis. Genetic diversity is important to organisms being able to survive through time. Okay, so last couple of slides, our general comparison. Meiosis and mitosis are both nuclear divisions. So we are dividing up the nuclear material, which in this case is going to be our DNA. Meiosis produces 4 daughter cells and mitosis only 2. And remember, the daughter cells from meiosis are only haploid. They just have one set of chromosomes for humans, 1 through 23. And in mitosis, they're going to be exact copies. So a diploid cell going through mitosis is going to make an exact copy that, again, is diploid. And for humans, that's 2 times 23 for 46. DNA replication is going to happen at the beginning of each of these divisions. Meiosis goes through those two sets of divisions, meiosis 1 and 2. Mitosis only has the one. Bam, and you're done. In meiosis, we know that each cell is genetically unique, period. And in mitosis, they are genetically identical. So here's a comparison side by side. If that helps you, you can pause the slide here if you'd like. Last couple of slides here. So we know that meiosis only occurs in reproductive organs. So it's not happening. It happens all the time, but puberty or later, essentially. For human females, meiosis isn't finished unless the egg is fertilized. So it may only happen a couple times in your life. So mitosis is very common, and it's going to happen in all your tissues for growth, repair, and healing. We know abnormal rates lead to cancer. Sorry to be doing this. Oh, we have to have another video to talk about non-disjunction. So I will end this video here, and I'll see you in part 3.